Okay, let's get started. Happy Friday. So Friday that we have class, that actually makes sense. I think is a good thing. Wanted to continue today with talking about promoters. So we're going to talk about transcription all day today. That's going to be DNA to RNA and how that process happens. One of the things that has to happen as far as transcription is you need to find the right place to start. And finding the right place to start is a promoter. These promoters are going to be sequences which I forgot to talk about last time, which are so-called upstream from where the transcription starts. Now, what do I mean by upstream? When you think about transcription, it's going to start at one particular position, and just like all of the other nucleotide polymerases, what direction is it going to go? 5' to 3'. So it starts here and then continues. One of the great things about these RNA polymerases is they can start in the absence of another primer. So they can literally start in one place. They start on one strand, which is also really important. And so with that strand, you know exactly where they start. If you stay on that same strand and then just count backwards, so instead of 5 to 3, which is the direction it's going to go from 3 to 5 back a certain number of nucleotides. And in this case, it's particularly the case where we have our -10 and -35 in bacteria relative to, again, where your first nucleotide is. You have sequences that bind to the sigma factor together with the RNA polymerase. And so those are the binding sites. That's where they are. They are 10 nucleotides away from where you start. That makes perfect sense if you think about it from a B-form DNA point of view, because 10 nucleotides away is going to be on the same side of your DNA. So it makes sense you're sitting down on one side of the DNA and can then start 10 nucleotides away from that. Also tells you it's a pretty big complex, because if it's 10 nucleotides away that it's binding and then starting right next to that, it's going to be a pretty large complex. So that's the concept then of a promoter. Then we have this concept of a consensus promoter, or what promoters are better or worse. And last time we talked about relative amounts of transcripts that you get. Some genes you have a lot of copies of RNA that come from it. Some genes you have very few copies of RNA. And to a great extent, that depends on how much RNA polymerase you have sitting to that promoter. So if you have more RNA polymerase sitting on one promoter, you're going to get more RNA from it. If you have less RNA polymerase, you're going to have less RNA being made from that. And one way for that to happen is just to have a particular sequence that the RNA polymerase likes to bind to, and other sequences that it doesn't like to bind to as much. And so just because of that, you're going to end up with more or less transcription taking place from that particular promoter. So one of the ways that you can figure this out, now as I've mentioned a few times, and I'll continue to mention for the rest of the course, it's actually relatively easy to sequence DNA. It's actually a lot harder to figure out where RNA starts. So if you just sequence the DNA and you look at that DNA sequence, just by staring at that sequence, you can get an idea whether this is likely to be a good place for RNA polymerase to bind, i.e. you're going to get a lot of transcription from there, or not such a good place for RNA polymerase to bind to it. How do you do that? You take all of the promoters that you can find, and some of them you have figured out exactly where they start, and then you literally line them up next to each other, right? One on top of the other. And then what you can do is look at those particular positions and see how frequently individual nucleotides show up. And that's what's shown here. This, I think, was a couple of hundred promoters that they're found in E. coli. And you find that around this -35 region, you have very often T's here, G's here, A's, C's, and A's here, but not all the time. You know, sometimes variations. And it turns out if you find the most common nucleotide at both of these positions, this -10 position is lots of A's and T's, which should sound really familiar in about 15 to 20 minutes, that these are now where you have binding by the holoenzyme. But if you have sequences that are very similar to the most common ones, you have really tight binding by the polymerase, which means you're going to get a lot of transcription from there. And if you have those that are different, you're likely to have a lot less which are being made from there. So the other thing, again, sort of pointed out at the beginning when I talked about upstream, people also talk about downstream, so after the transcriptional start site, and you're always talking about the DNA in this particular case. And what that says is that these are always going to be directional. So if you have a particular piece of DNA and 10 nucleotides and 30 nucleotides away from that particular position on one strand, then it's likely to be transcribing in this case, I'll have the down direction. If, however, you have a sequence here and on the other strand, sequences minus 10 and minus 35, you'll be transcribing the other strand. These are going to be completely different sequences because they're just complementary to each other. So you're going to end up with very different sequences, very different proteins in the actual process. So now we're all warmed up. Most people made it. Let's move on to a clicker question because they're so much fun. Yes? Well, we can go back. I can go back. No one read that unless you're really quick. Yeah?
You were talking about the amount of RNA polymerase you have determining the amount of transcription.
Mm-hmm.
You're saying if you have a lot. Are we talking about different promoters for the same gene? So like that gene sequence at different places on the genome. Are we talking about how many RNA polymerase molecules you have binding at all of those promoters for the same gene? Or are you talking about multiple RNA polymerase molecules at the same promoter simultaneously?
Okay, so I'll try and clarify the question here too. Yeah, but the basic question is really, to some extent, do you have multiple promoters, i.e. binding sites for polymerase, for one gene? Or do you just have a single promoter for one gene? And the answer is, at least in bacteria, it's pretty much you have a single promoter. But if you have good binding to that promoter, you'll have to get the polymerase that was bound there to move off before you have another one that binds to it, because these are specific sequences that's going to happen. If you have a strong promoter, that's a specific sequence. And so it means it binds well. So as soon as one polymerase is left, a new polymerase will come down and bind to it. Whereas if you have a weak promoter, the polymerase will come down and bind, it'll take off and do its thing, but it'll be a long time before another polymerase comes down and binds there. So it's really about how frequently you have the polymerase binding to that particular promoter. So it's not simultaneous that they're all bound at the same time. It's a series, the one after another. That make more sense? Yeah. Okay. Good. So now we can do our clicker question. And it will say that we need to do our base frequencies, etc. Good, we actually have the numbers up here. When a conserved DNA sequence, is found in multiple related organisms, the most likely interpretation is the DNA is transcribed, is functional, is a promoter, is not a promoter, encodes a non-coding RNA. I'm going to try to. Do we want to restart on the time? As soon as I can move it, I'll restart it. Let's do this. Why is it not letting me move it? Because it's probably yes. Let's just stop and restart, okay? Yes? Stop. Move. Up. Hopefully this will work better. Go back to present. Play. Okay, that better? Good. Okay. When a conserved DNA sequence is found in multiple related organisms, the most likely interpretation is transcribed, functional, promoter, not a promoter, encodes a non-coding RNA. And I'll usually try not to lecture that fast. 5. Okay, nobody liked my not a promoter and non-coding RNA, or very few people like those. Not a promoter, actually, you really can't tell because as we just talked about here, there's a lot of variability. You can have a really weak promoter that is not going to be very close to your consensus sequence, so it's very hard to tell what it is. So C and D are out. Actually, E is not a bad answer because non-coding RNAs are often sequences which are conserved in multiple different organisms, but you can't really tell. And if you think about what other sequences might be there, transcription is certainly a possibility. Functional actually gets transcription and a possible non-coding RNA. So the most likely interpretation, and people always hate me when I write these kinds of questions, is that the DNA is functional. So it's a functional piece of DNA. It says most. Most. Okay, so this is just a reminder about the fact that you can look at upstream and downstream sequences, and depending on where your promoter is going to determine what kind of sequence ends up being transcribed. So here, for instance, if your promoters over here on this strand, your -10, -35 are here, you're going to make an RNA, in this case, that's all C's. If, however, your promoter sequence is over here, the polymerase is going to transcribe. This is exactly the same sequence, same DNA, but if the polymerase is going this way, you're going to make one sequence. If it's going that way, you're going to make another sequence. And this is actually pretty clearly shown. All of you remember, of course, the E. coli genome. We talked about like forever ago before the last midterm. That there are lots of genes on the different strands of RNA. And there's no sort of rhyme or reason to why one strand is used for coding versus the other strand being used for coding. And in this example down here, all the little green arrows are promoters. So sometimes you'll have two promoters that sit right next to each other, but they're going to direct transcription in opposite directions. You remember that TA-TA sequence? If you think about TA-TA, that's actually going to be really similar on the opposite strand, right? So that TA-TA in and of itself is not going to tell you whether you're going to be transcribing this way or that way. So you need some of those extra sequences, and the -35 sequences are actually very different if you look at them on the opposite two strands. But that's by no means the only kind of situation you get with promoters. Quite often you'll have just a promoter pointing in one direction. But the take-home message here is that you've got two different strands, which are going to give you two different sets of genetic information. And it really depends what the promoter sequence is and where that is relative to your transcriptional start site. So once you've started transcription, then you have all of these cool conformational changes. We talked last time about abort of initiation that sticks out a couple of nucleotides before you continue to move on. But there's a big conformational change that happens once you're actually undergoing elongation. And the main thing there is that this becomes incredibly precessive. It does not fall off of the template until it will get to a terminator that we'll talk about in just a second here. Yeah?
When there are sequencings, like an organism's genome,
Yeah.
are they doing just one strand or is it both strands?
Okay, so the question here is when someone's sequencing a genome, are they doing one strand or are they doing both strands? We'll talk a little bit about sequencing technologies right at the very end of the class, but the great thing about sequencing and the fact that DNA is complementary, if you sequence one strand, you know what the sequence is on the other strand.
Sure, yeah. So what do they just like, when you see the list of bases, do they just like snap the other side onto You know what I'm saying?
Yeah, so when you're looking at a genome sequence, they usually only show one strand. But then the other strand is assumed as being complementary to that. I don't know if that answers your question.
Well, I think what I'm thinking is like if you say that you have 50,000 base pairs.
Yeah.
I mean, the reality is you have double that's potential for gene coding, right? Because both ends are encoded
Yeah.
for separate genes.
Yeah, so that's exactly right. So I'll just repeat this. But yeah, if you've got 50,000 base pairs on one strand versus another, you've actually got 100,000 base pairs of potential information that you could transcribe into. That's exactly correct. So knowing which strand is actually being used is really, really important because it's going to generate something which is really different. When we talk about the genetic code next week, that turns out to be really different as well. The other thing is where you start turns out to be really important too in terms of what protein you end up with. Yeah?
So say, like you pointed out on the last slide,
Yeah.
the promoters are at the same spot on
Mhm.
the DNA strand, and so they are going to transcribe from exactly the same spot.
Oh, sorry. In this case, the bottom here? Yeah.
Would, if they end up doing either side of the same spot of the DNA, would those RNA complexes they end up with encode for different enzymes or proteins and genes, even though they're complementary sequences?
Yeah, so the question here basically is if you do transcribe, and again, just like it's shown up here, this is, of course, an oversimplification, but you've transcribed the same piece of DNA right here. Here's a transcribed piece of DNA, the same piece of DNA here. This information is completely different than that information. So those two different strands are actually encoding completely different information. And then when we talk about the genetic code, it turns out that each of those can encode 3 different proteins. So yeah, it's a very compressed amount of information which is in there. Yeah, it's really long and a whole bunch of non-coding sequences that are present in our genome, but the bits that are coding can actually encode a lot of information. Yeah, so this slide is basically just to show 2 things. One is that this is your elongating RNA polymerase, and this particular piece sort of looping over here into the gap where the 2 strands have been pulled apart, that's what's really giving you processivity, because it's really looped around. It's actually a lot like we'd have for a helicase in terms of looping around the DNA. It's not actually covalently closed, but it's providing this processivity. The other thing which is important here is that you might think it looks like a hand, but relative to all of the DNA polymerase structures, it's completely different in terms of how it all comes together. The other thing that they're trying to point out here is we know a lot about the high-resolution structure of these DNA-dependent RNA polymerases. A couple of things that we got, in fact, from those structures are that, we have a very short piece of DNA and RNA that are bound to each other. You can see that in cartoon form over here, but actually this is a real high-resolution structure of one of these elongating polymerases, and that it does wrap around both double-stranded DNA and each of these single-stranded bits. And this RNA comes off of that DNA very, very quickly. And this was a big argument even back in the late dark ages when I was in graduate school, is how long that particular piece was. Yeah?
So is this, are the different colors representing different domains on the polymerase, or is this the polymerase and then accessory proteins?
Okay, so the question here, again, just to repeat it, is basically what are these different colors here? Not so much the colors, of course, the DNA. Your RNA in orange, the DNA, the 2 different strands in yellow and red. But the different pieces that are on here, these are actually different subunits of the polymerase. So they're different pieces that come together. In the case of the bacterial polymerase, we'll see in just a second, there are relatively few of these. In the case of the eukaryotic polymerases, there are a lot more. Yeah?
The RNA polymerase is what separates the DNA strands to allow RNA transcription, correct?
Yeah, so the question is basically, sorry, I'll paraphrase here, but does the RNA polymerase need a helicase like DNA polymerase needs a helicase? And the answer is no. At least the bacterial enzyme, and we'll see there's actually a slight difference for the eukaryotic enzyme. The bacterial enzyme is its own helicase. It pulls the two strands apart by itself. Okay, so this is your elongating polymerase. It's clamped down. It's got a little short piece of the DNA attached to the RNA. This is great, but there's also an end to a gene. You don't want this thing to go all the way around your genome. So how does that stop? Turns out it's relatively well known in E. coli how this happens. It's actually not very well known in eukaryotic cells how this actually happens. But in bacterial cells, there are really two different kinds of termination that happen. So termination is just the end of transcription. You had your initiation, get to the end, termination. And there are two ways that this can happen. One is a protein-dependent process that actually depends on having a specific protein bind to the polymerase and the RNA and pull that RNA out of this elongating complex. But there are also protein-independent methods, and this is the easiest one to understand, which is here, where some people, actually one of my students a couple of years ago called this, speed bump and oil. So the idea, the speed bump here is, back up, a RNA structure which forms in the RNA as it's being made and forms this stem loop structure where the newly made RNA can bind to itself, and then the oil is this whole stretch of U's. Why a whole stretch of U's? Because U's and A's only make 2 hydrogen bonds as opposed to G's and C's, which are going to make 3. So it's a relatively weak interaction. The other thing here is that, as I was trying to point out previously, is that here it's got, I guess, like 8 base pairs. That's not the case when you have an elongating polymerase. You just have a couple of base pairs, which is there. And so forming this secondary structure and having a couple of U's right next to it basically pops the RNA off of the polymerase. Once that RNA has come off of the polymerase, the two strands come back together, and then the polymerase falls off of your DNA. It will go back and try and find another promoter. And again, depending on whether it's a strong promoter or a weak promoter as far as that happens. So what happens, again, it's really about the RNA structure here, and that RNA structure basically pulls the RNA out of your elongating complex. Yeah?
This might have been covered before, but is there anything that stops another polymerase from coming on and from starting again? And starting the process over?
Okay, so I guess the question here is there anything that's stopping the polymerase from coming in and starting again? At the promoter, no. As long as if there's no polymerase there, you'll have another polymerase that can bind there. If, however, you're not at a promoter, so you don't have that particular sequence, then the polymerase is not going to bind there. So for instance, here, when you have the polymerase which is dissociated after the termination, you're not going to have another polymerase bind there unless there's a promoter sequence, which is right there, which is highly rare. And also, if you've got this stretch of U residues, all remember your -10, -35 sequences. I'm not going to ask them on the midterm. But they're not a whole stretch of U's. So that way, that's not going to be a particular promoter sequence. Now, it's not uncommon that terminators and promoters are right next to each other, but that's a slightly different story. Okay, so that's bacteria. Bacteria are nice and simple and easy to work with and wonderful system. Yes?
I'm sorry, you said that bacteria does it 2 ways, didn't you? It has 2 ways of termination?
It does have 2 ways of termination. I just briefly mentioned the one which is protein dependent. It's also called row-dependent termination because that's what the protein is called. But we are going to concentrate on this particular one because it's a lot easier to follow. If you'd like, we can talk a bunch about row-dependent termination, but it's a fascinating system, really cool. But yeah, there's so much we could talk about in this class. Yeah, so switching over to eukaryotes. Bacteria are a wonderful system. There's only one DNA-dependent RNA polymerase, very specific promoters it associates with. We know a lot about how it works. Eukaryotes are a little more confusing. they don't have just one DNA-dependent RNA polymerase. They have 3 DNA-dependent RNA polymerases. And again, as our friends the molecular biologists are wont to do, incredibly creative names, 1, 2, and 3. They are going to transcribe different sets of genes. So RNA polymerase 1, for the most part, transcribes ribosomal RNA genes. Now, why is this highly likely that you have your own polymerase for doing these things? Anybody remember what the most common RNA is in the cell? It's your ribosomal RNA. So it's not terribly surprising that you have your own polymerase, because it's really important that you make all of these ribosomes, and you make a whole bunch of your ribosomal RNA. So RNA polymerase 2 is the one that transcribes all of the protein coding genes, and we're mostly going to concentrate on RNA polymerase 2, because also it's been best studied. There's also an RNA polymerase 3 that transcribes some ribosomal RNA genes, but is mostly important for tRNA genes. Again, the second most common ones that you have present in the genome. Yeah?
Is there any difference in fidelity between any of these 3?
So difference in fidelity between RNA polymerases 1, 2, and 3, not that I know of. One of the things that I think I mentioned last time, but if I didn't, I can mention again now, is that the RNA polymerases don't have as high fidelity as DNA polymerases. And the reasoning behind that is if you make one mistake, it's not going to get copied again and again and again. Whereas for DNA polymerase, that is the case. And that's probably the trade-off that you have. You can start without a primer, but you have low fidelity. So that seems to be the reasoning behind that. So these 3 RNA polymerases were almost definitely derived from one ancestral RNA polymerase and have then diverged in these organisms. What does that mean that these things are relative to each other? Orthologs, paralogs? Same organism means? Yeah, so these are going to be paralogous genes in this particular organism. And as often with paralogs, they're going to have slightly different functions. Again, transcribing slightly different genes. Multi-subunit, I mentioned before, it actually great lead-in on that figure before. 5 subunits, 5 different proteins that come together to make the bacterial RNA polymerase, but lots and lots of them for the eukaryotic RNA polymerases. When I say lots and lots, it turns out it's the variability between polymerases 1, 2, and 3 in terms of which ones are there. So this is a way of looking at that. This is a table that was put together by my colleague Mike Bartlett, who actually works on some of these polymerases, actually literally archaeal polymerase over here. But basically, of the 5 subunits that you have in the bacterial polymerase, each of these has a homolog in the eukaryotic polymerase, but the eukaryotic polymerase also has a few extra ones, like these 4, 5, and 6. RP, by the way, here is RNA polymerase. B are the genes that correspond to 1, 2, or 3. So if it was RPA, that would be from RNA polymerase 1. RPB is from RNA polymerase 2. RPC is from RNA polymerase 3. So just to give you some terminology there. Why they called them beta, beta prime, alpha, etc., don't ask. I'm not sure where that came from originally. But beta and beta prime and RPB1, RPB2 are the 2 major largest subunits which also have the enzymatic activity. So they're the main things which are happening here. Alpha 1 and alpha 2, we'll see these guys later on when we talk about regulation. Turns out they're really important for regulatory aspects of these RNA polymerases. So that's basically, again, what it says at the top here. They're all related to each other. But what should be really fascinating and amazing, and all of you've noticed, is what's happening over here in this column. Is that the archaeal RNA polymerases actually have a lot more similar subunits to the eukaryotic polymerases than they do to the bacterial polymerases. And it's not terribly surprising then, if they've got really similar subunits, they actually also have extremely similar structures. And so these are high-resolution crystal structures of the bacterial polymerases, archaeal polymerases, and eukaryotic polymerases. When I say this, of course, bacterial, archaeal, and eukaryotic, that actually means this is from, I think this was from Thermus thermophilus, this one's from Sulfolobus sulfatericus, and this one's from yeast. So, but it turns out that those are extremely similar to each other inside each of those individual domains of life, and so similar structures, likely to be similar functions, and also probably something that was present down here before some of the dispersion of all of these different domains of life. Structures, very similar. Coloring here in a very similar way as we had on that last table. The gray and gold, I guess, here, those are the two largest subunits. That's really the business end of the polymerase. And it also has this really kind of claw-like structure. That's what's grabbing the double-stranded DNA. And the active site is right down here in the middle. True for all of these guys. So what about RNA polymerase II? This is now RNA polymerase is making all of the messenger RNAs. Unlike bacteria, it doesn't have a sigma factor. So how does it find the right promoter? Well, it turns out it has analogous but not homologous proteins for doing this. Not homologous means there's no sequence similarity, probably not derived from the same common ancestors. So instead of this sigma factor telling you where to start, where the promoter is, it uses all of these so-called general transcription factors. General transcription factors, alphabet soup, unfortunately, known as TFs for transcription factor, 2 because it's RNA polymerase II, and then A, B, D, E, F, H, etc. We're going to concentrate on B, D, and H for this course, even though a really good friend of mine spent 5 years of her life trying to figure out what the sequence was of this particular one. But that's the way that the RNA polymerase II could find promoters, find the right place to start. The other issue that, of course, eukaryotic polymerases have to deal with is there are a bunch of nucleosomes bound to your DNA. And, as we'll see, the RNA polymerase, relative to the DNA, is pretty darn big. So getting to the promoter, getting to the right place, is actually really kind of problematic in eukaryotic DNA. And we'll see that it's not just these general transcription factors. It turns out you need a lot of the other chromatin remodeling complexes that we talked about before when we were talking about regulation as far as histone modification. So what do these general transcription factors do? They find the promoters, probably the main one that they do. What do promoters look like in eukaryotic RNA polymerase II promoters? Tata box. Should sound kind of familiar, and kind of like that -10 sequence. So Tata box, boxes. When people talk about boxes, and I'll probably spend too much time talking about boxes here as well. A box is just a conserved DNA sequence. Some people, at least originally, called the -10, -35 sequences the Pribno box. So those are, again, these conserved DNA sequences, and I think the reason is people just put boxes around them, like when we talked about way back at the conservation very early on, just the box around it. That's why these are called boxes. Tata, again, not surprisingly, because it's got a rich in TA sequences. It's about 25 nucleotides upstream of the transcriptional start site. So you've got this Tata box in your DNA, start a transcription, again, about 25 nucleotides further along. How do you get the RNA polymerase to here? Well, the first thing that happens is you have TF2D that associates with this Tata box. Now, how does it associate with it? Through the, again, incredibly creatively named Tata box binding protein, also known as TBP. So TBP is part of TF2D. TF2D is a complex of multiple proteins. Then, once you have TF2D binding, then you have TF2B binding. In some cases TF2A, but TF2A is not required for everything, so you'll notice it's not here on the slide. TF2B binds to both TF2D, but also a very specific sequence, which we could also call a box, but people named it something else. They called it an element. So element is a DNA sequence, a box is a DNA sequence. So the TF2B recognition element, the BRE. So TF2B interacts with the BRE, Once you have these two complexes of protein bound, then you have TF2E, TF2F, most importantly TF2H, that now associate with the RNA polymerase and bring this whole thing to the promoter. Bunch of different proteins, whole thing that has to be added together. TF2H has a couple of interesting activities. One, it's a helicase, and that's the easy way to remember it. It's an H for helicase. And that tells you one of the differences between RNA polymerase II and the bacterial RNA polymerase that you asked about before. The RNA polymerase II needs another helicase activity. It can't do the job without TF2H, unlike, again, the bacterial polymerase. The other activity that TF2H has is a kinase activity. What do kinases do? They phosphorylate things. What gets phosphorylated in this case? What gets phosphorylated is the RNA polymerase itself. So the RNA polymerase has this thing called the C-terminal domain, or CTD, that gets heavily phosphorylated by TF2H. And part of that phosphorylation actually seems to be not unlike what we saw with modifications in histones. Negative charges are pushing away from the DNA. This phosphorylation seems to help with getting that RNA polymerase away from the particular promoter. So this is basically said again in words, what I just showed on that last slide. TF2D has the TATA binding protein, and then, unfortunately named, TATA binding protein associated factors. Factor is now a protein. So elements and boxes are DNA, factors are proteins. Again, I didn't make up this terminology. It's not my fault. So the TATA binding protein associated factors, these are the rest of TF2D. And it turns out that these are really important for the regulatory functions that we'll talk about after the next midterm, so you'll remember them again then. TF2B recognizes this TF2B recognition element, and importantly, we'll take a look at this in just a second, TF2B is on one side of your TATA box and not on the other side of your TATA box. And so this gives you the directionality of which direction your polymerase is going to go. Because TATA, if you just say look at TATA, TATA on one strand is actually identical to TATA on the opposite strand. So just the TATA box doesn't tell you what direction, which strand you're going to be using for transcription. Having a BRE, however, will let you do that. We'll ignore TF2F and TF2E as far as this course is concerned. TF2H, on the other hand, has the helicase activity and the kinase activity. Okay, questions about these individual ones? Again, I'm not going to ask you about E and F. Got enough alphabet stuff to worry about already. Okay, so what are we looking at here? TATA box we already talked about. Here it says it's minus 30. In some cases it says it's minus 25. Turns out it's actually pretty variable. Unlike in bacteria, where you have a really clear minus 10 and minus 35, the TATA box can be a bunch of different places here. The TF2B recognition element can be in a bunch of different places as well. And what this means is just at looking at DNA sequence, like we were talking about before, it's actually really hard to tell where these promoters actually are. So just staring at the sequence is not necessarily going to tell you that. A couple things to mention here as well. These are the two most important DNA sequences. There are some sequences at the what are called initiator element and downstream promoter element, which in some promoters seem to be important for the association of the RNA polymerase to the promoter, but certainly not all. Some promoters actually don't even have TATA boxes either. But these are the most important ones, the BRE and the TATA. If you look at the TATA sequence, this is the consensus sequence. Again, not unlike the consensus sequence we looked at right at the beginning of lecture today, where we saw how frequently you see any of these things. One thing to mention here is that in the initiator element, you'll notice C or T. So you find C or T just about as often as anything else. N, etc. Lots of things that are really not very well conserved here. Same thing is true on the downstream promoter element. One thing you might actually notice here, though, is that TF2D is interacting with this INR and this DPE. But wait a minute. TF2D is bound to the TATA box way the heck over here. How the heck do you get from 30 nucleotides upstream to 30 nucleotides downstream? How do you do that? You bend the DNA, which is actually exactly what TBP does. So TBP, when it binds to DNA, it binds, interestingly enough, mostly in the minor groove rather than the major groove of DNA. And in that process, bends the DNA, and a really sharp bend that happens in the DNA with this DNA binding. And just as an interesting aside, I didn't have a chance to go and find this in my office. 1992, telling you a little bit about how old I was. This is second year in graduate school. People were really excited about these structures and figuring out exactly what the structure looks like. The first actual structure of the TATA binding protein was in the absence of DNA, and literally on the cover of Nature, they had a double-stranded DNA, no bend whatsoever, right in the middle, right here. So it was completely wrong in terms of what the DNA looked like. And then about 6 months later, they came out with this structure that actually had the DNA in the right place in it. So it tells you don't have to actually be right to be on the cover of Nature. So this is that particular, and actually the double-stranded DNA fits perfectly right down the middle here. So, if you were just guessing, oh sure, it's a great place to put the DNA. But no, it makes this very sharp bend in the DNA. And that's that bend which is really important for getting all of those other factors together. The TBP together with all the rest of the TF2D complexes, TBP, ENF, H, and the RNA polymerase. So all of these guys come together to make the so-called pre-initiation complex. We haven't even started to transcribe yet. We've got all of these proteins associating with each other. Here's the cartoon form. There's your TF2D. Here's the polymerase. Here's your TF2H getting ready to phosphorylate here. There's this big purple thing. What's this big purple thing? It's called the mediator complex. Mediator is probably anywhere from 20 to 30 different proteins. That seems to be important for most promoters. As we mentioned before, this has to happen in the presence of histones bound to the DNA. So you've almost always got histone modification enzymes, chromatin remodeling enzymes. So all of this stuff has to come together in order to get transcription to take place. It's really kind of amazing that we're alive at all, considering that you have to have all these things associated with the promoter until you actually get transcription to start. Yes?
Are we going to go in and dissect the mediator at all, or are we just going to kind of think of it as this nice purple blob?
Are we going to go in and dissect the mediator at all? Is it going to be a big purple blob? We are not. It's just going to be a big purple blob because, again, it's 20 to 30 different proteins, and we don't know what a lot of them do. There's actually been some arguments in the literature how important mediator really is. So come back, take this course in 5 years. We may spend a lot more time on mediator, but not this term. So it's a blob. But the important thing is that the blob is not only interacting with all of our general transcription factors, it also interacts with these specific transcription factors. Here, activator proteins. And we'll talk a lot more about activator proteins later on. The mediator seems to mediate the activity between these activator proteins and the general transcription factors. And that's basically all we need to know about the mediator. So once this happens, once you have, in most cases, activity of one of these activator proteins, then you can get transcription to take place. And once transcription starts taking place, the TF2H will pull the two strands apart. The RNA polymerase will do a little bit of abortive initiation. Then once you get the kinase phosphorylating appropriately in the C-terminal domain, then it'll take off and be highly processive until it gets to the end of the gene. Unless, of course, you run into the same nasty problem that we talked about with DNA replication, which is when you pull two strands apart, you actually start to generate supercoils. in front of, and actually also behind, because you're putting that DNA back together, winding and unwinding the DNA. So you also have to have topoisomerases in order to help out with this supercoiling that happens. So yeah, no, we don't have enough with all of these pre-initiation factors. You need topoisomerases as well. Turns out that RNA polymerases don't just get on the DNA and cruise all the way through at the same speed. It turns out that there's a lot of stopping, starting, slowing down, speeding up, etc. And there are a number of factors. Factor means what? Proteins that are also involved in how fast the polymerase is actually moving along this whole process. One of the things, however, that's probably good about the supercoiling that's generated when these things are being pulled apart, you pull them apart, in front of the polymerase you're generating positive supercoils. Why is positive supercoiling useful? Because it helps actually to remove nucleosomes from your template. So just the fact that you're pulling apart the two strands helps pop nucleosomes off of the DNA so that you can have transcription come through there. And this, again, the polymerase relative to the DNA is really big. And there's no way with that, wrapping around one of those strands that you could still have a nucleosome associated with your DNA and still get transcription to take place. So the removal of nucleosomes is also a very important aspect of this pulling apart and the whole thing coming back together. Any more questions on this? Yes, back.
So when the RNA polymerase is paused waiting for nucleosomes to get unwrapped, DNA unwrapped from nucleosomes, and those proteins or the RNA sequences are hanging out outside the polymerase, are those subject to degradation or damage while they're waiting for the nucleosomes to unwrap?
So I'll paraphrase your question a little bit and tell me if I'm missing it. But the question is about what happens to the RNA when it's hanging out off of the polymerase. What's happening to it basically as it's hanging out there? The answer is there's a lot of processing, there's a lot of capping, eventually tailing, but also splicing that's going on in that case. But there's nothing particular that's protecting it in that case. In the eukaryotic case. In the eukaryotic case, but all of that's happening then in the nucleus. And the nucleus is probably a relatively protected place. On the other hand, if you think about bacterial transcription, the RNA as it's coming out of the polymerase actually gets translated almost immediately. So that RNA comes off, the ribosome associates with it, and it starts getting translated. So that's a little bit of a different thing that's going on in bacteria versus in eukaryotes. But in eukaryotes, that RNA just seems to be hanging out there. And having a pause doesn't seem to make a difference. There doesn't seem to be too much degradation that takes place there. Hey. Good. No more questions? And I have a question for you. Which of the following bind to specific DNA sequences? TF2, B, D, or H? Or some combination thereof? Ten, 5, 1, 0. What do we think? We think D. What do I think? I think D. So, what does TF2B bind to? TF2B binds to the TF2B binds to the BRE. TF2D, through its subunit, the TBP, binds to the Tata box. TF2H binds to all those other proteins. So not to the DNA. So D is correct. Okay, one of the things that I've mentioned a few times in this class is that anytime you have a textbook, it's going to be out of date. This is a publication that came out about 2 years ago now, looking at this whole complex and trying to piece apart where all of these proteins are. This is a combination of X-ray crystallography and electron microscopy, and we'll talk more about that a little bit later on. But if you just look at this, and it's not, the details here are really not important. TBP is part of the TF2D complexes, bends the DNA really sharply. TF2E is present in the middle here. This, the tail module, this head module, that's that big purple thing that all we need to know about is it's a big purple thing, only here it's blue. Here's the mediator complex, all associated with the RNA polymerase. The RNA polymerase is here in gray. Here's TF2B binding to the BRE right next to the Tata box. And then we've got, TF2H, all of these other things in here. So again, it's a really ginormous complex that's all sitting down on the DNA. And to get all of that there, clearly you need to have a lot of stuff removed in order to have this particularly take place. And partly because of that, we've looked for simpler systems to try and understand what's going on here. You remember that table where we had the subunits from bacteria, eukaryotes, at least RNA polymerase II, and archaeal polymerases? Archaea are a really wonderful system for studying transcription because, A, they have one RNA polymerase instead of 3 RNA polymerases. How that RNA polymerase deals with transcribing ribosomal RNA genes and tRNA genes is a very interesting question we don't know the answer to. But that RNA polymerase is very similar to RNA polymerase II. It also has general transcription factors that are important for its function, but it's only got 2. And they don't have a TF2D, it just has TBP and TFB. And this is called TFB rather than TF2B because there's only one polymerase. So you don't need to give it 1, 2, or 3 in terms of names here. And so this process, TBP binds to a Tata box, which is very similar between RNA polymerase II promoters and archaeal promoters. TFB binds to the TFB recognition element, which is very similar to the TF2B recognition element. And then the RNA polymerase comes in. You don't seem to need any of those other extra factors. And the RNA polymerase in and of itself has helicase activity, so it's not like needing this extra TF2H. And, excuse me, this can then start transcription. So it's a nice, much simpler system for trying to understand the basics, particularly of the RNA polymerase and how the RNA polymerase works. So this is what we keep telling all the granting agencies and telling them they need to give us huge amounts of money so we can study this. It hasn't worked terribly well yet. But what we know from transcriptional initiation in archaea is the polymerase, here now in purple, sorry about the not keeping the color schemes the same here, is extremely similar between these whole groups of organisms. TFB is very similar to TF2B. TBP is very similar to TBP, but you don't have all these extra TAFs. You don't have all the TF2H, TF2E, etc. So it's a nice, simple system of trying to understand, again, the very basics of what's going on here, and it's probably more similar to the ancestor of all of the currently 3 RNA polymerases that we have in eukaryotic cells. And to sort of to not hopefully beat the horse too much here, these are just a couple of X-ray crystal structures looking at TBP in both a archaeal system and a eukaryotic system, and TFB and TF2B in an archaeal and a eukaryotic system. Who cares about this particular eukaryote? This is the human system here. TBP, practically identical in terms of the sequence, the structure, how it interacts with the DNA. TF2B interacting here with the BRE is, yeah, it's a different color, sure. But other than that, these structures are basically almost completely superimposable relative to each other. So, great model system for trying to understand what's going on here. There are a couple of differences between the archaeal polymerases and the RNA polymerase II. One of those is the C-terminal domain, CTD. What happens to the CTD? Gets phosphorylated by TF2H, exactly. So there's no TF2H in archaea, so it's not surprising there's no CTD in archaea. Initiation factors, you've got a whole bunch more in the RNA polymerase II system, none in archaea, although people argue about this. There may be a TF2E homologue, but as far as we're concerned, they're not there. What about the DNA? Turns out that in eukaryotes, we've got nucleosomes, we've got histones, we've got all kinds of modification complexes. There are a few histone-like sequences that are present in some archaea, but certainly not all of them. Definitely the DNA needs to be packaged. You have packaging, same kind of thing that you have to have with bacteria. But it's a lot less well understood exactly how that's working. So the last thing that I wanted to do was to show you this transcription movie. And we'll see if I can do this without the sound completely freaking out on me here. This is a nice animation from the DNA Learning Center. This is now eukaryotic transcriptional initiation. We have TF2D here. So the TBP is down here at the bottom, binding to the Tata box and giving you a nice bend to the DNA. Then you got the rest of TF2D sort of hanging out and flopping around here. So that is now waiting for what? TF2B, exactly. So TF2B needs to come in, bind to this bent DNA sequence, interact with the BRE, and say, hey, this is the direction that we want transcription to take place. There it is, together with the polymerase. You've got a couple of other subunits. There's the mediator, unfortunately not purple, but in this case green. Now everybody's here, everybody's sitting at the promoter waiting for that activator protein to come in and basically kick off the whole process. Once that happens, you have your RNA polymerase. It's now split the two strands here. Here's the RNA coming out of the top here. The sound effects here are really cool, but I decided not to put them in. Racing along in a highly processive manner, incorporating into these individual ribonucleotides into this growing chain. And once this gets to a termination point, then the RNA will be released. We'll talk about what happens to that RNA next week. And this is that whole process here. If you would like, we can go over it again, or we can call it a weekend. Call it okay, quick question here before we all call it a weekend.
Back on Dr. Bartlett's image of the transcription initiation archaea, it looks like it had a phosphorylated tail on the bottom of it, but if it doesn't have TB2H, what's phosphorylated?
Okay, so the question here had to do with one of the previous slides, so we'll go back here to Keynote really quickly and play here in this case here. Okay, so it may look a little bit like there's a tail in this case. The red is supposed to be the RNA transcript. So actually, no, there is no phosphorylation that is taking place on any equivalent of the C-terminal domain in this case. So we'll talk about RNA modification on Monday. Have a good weekend.